So Rostislav, just to ask, the session is recorded, if I understand correctly, and the recordings will be put online later.
That's great.
Yes, exactly. So we have started recording, but we still have a minute or so before the official start. So, but whenever you feel, you can.
Okay.
Sorry.
Let's wait for the official time. And
Yes.
I just wanted to turn on camera, but Zoom is against me.
But don't worry, I think we have time. So, we can even have a few minutes, be a few minutes late. I will time it so you can disconnect and try to connect again if you want to. I think it'd be good if, the camera works.
So let me probably, Joseph, introduce you. So we have our welcome everyone to our second session of the ELLIS Lightning Talks of this ICTERI-2024 conference. And I have a great pleasure to introduce our chairman of the next session. This section was co-organized with Professor Josef Šivic from Czech Technical University in Prague. And Josef will be a chairman for this session.
Okay. Okay, I could not turn. Sorry, I could not turn. Okay, I could not turn a camera on a laptop, so I instead turn it on an iPhone. So if you see iPhone micro, that's my head, and
perfect.
Yeah, that works. I think you can maybe try sharing your screen and then.
Let's start with that. Shares. Okay. Hope that you see it.
Yes.
Reasonably well.
I see it well.
And do you see my mouse?
I see the mouse. Do you want to try any video before I introduce you?
Yes, we can try a video.
Seems to work perfect, yeah. So
great, maybe this one.
And you will have
protest. Yeah, but it works. Okay, great.
Yeah, and you will have, so this format is 20-minute talk plus 5-minute question session. So
that's great.
Okay, so if okay, I will then. So Rostislav, can we start? Can I introduce?
Yes, please go ahead.
Okay.
You are in charge now. Whatever you want to do, you are free to do.
Okay, thank you very much, Rostislav. So it's really a great pleasure to be here. My name is Josef Šivic. I'm chairing this session. So welcome to the ELLIS Lightning Talk session at the ICTERI conference. This is a session jointly organized by the ELLIS Associate Unit Lviv and the ELLIS unit in Prague. And it's really a session focusing on presenting and discussing recently published results in machine learning driven fields. And so this is actually a second session. The first session was earlier today, which was a very exciting session. And in this afternoon session, we have also a very exciting lineup of 3 speakers. The first speaker is Michal Neoral, who is a PhD student at the Czech Technical University in Prague at the Faculty of Electrical Engineering at the Center of Machine Perception. He's an expert on visual tracking, and he's going to tell us more about his recent work on long-term tracking of every pixel. His work was published at VACVI, which is IEEE CVF Winter Conference on Applications of Computer Vision, and it was published, I think, this year. So, Michale, it's great to have you here, and I'm really eager to learn about your exciting work. The stage is yours. Thank you very much for joining us.
Thank you. Thank you for the introduction, and thank you for the opportunity to present at the Lightning Talks. I would like to also thanks to my colleagues Jonas Scherich and Jiří Matas, which are co-authors of the MFT paper. So this presentation is focused on a specific topic, which is a long-term pixel tracking, which is currently very hot topic and in interest of the biggest AI research teams in the world. So let's start with a brief introduction to some key computer vision terms to help everyone, including these outside this discipline, better understand the rest of the presentation. So let's start. In essence, the feature matching task or correspondence estimation means that for a point in one image, you want to find a correspondence position in another image. Typically, the feature matching methods estimate the position for a small number of points, significantly fewer than the total number of pixels in an image. So next level of this is optical flow, which has a task of estimating correspondences, but for all pixels from a reference frame to a target frame. However, this estimation is, as I said, performed only between two consecutive images, and optical flows are trained for small changes in images, typically at least. So on the other hand, long-term pixel tracking is not limited by the number of frames, and it estimates not only the position from initial frame to last frame or target frame, but also the positions from for all intermediate frames, and moreover, it also determines whenever the points remains visible in specific frames. So we are doing that one. So let's start with a paper. So our paper deals with visual tracking, long-term visual tracking. Standard visual object trackers tracked an object from the first video frame throughout the video using a bounding box or a segmentation mask representation. As you could see in the previous presentations. Applications like structure from motion or video editing benefits from long-term dense correspondences. So optical flow methods output dense correspondences, as I said, however, only between pair of frames. And our method called MFT, which stands for multi-flow long-term tracker, extends this optical flow to dense long-term trajectories over the whole video sequence. So for each point in the first frame, MFT outputs the points and visibility, positions as visibility in every other frame of the input video. So a baseline approach for this is simply to chain optical flow fields computed in consecutive frames or between consecutive frames. This is a toy example of that chaining, and the x-axis represents the number of frames or frame number, and the y-axis is the position of a tracked point. For simplicity, the diagram shows just one-dimensional case. As shown in the video, such approach cannot recover from temporary occlusion. Even if the pixel is occluded for one frame, this approach cannot recover from that. And even for a non-occluded areas, small errors in optical flow can accumulate over time, which leads to a significant drift from actual position. So another baseline, or another approach, is to estimate optical flow directly between a reference frame and target frame or a current frame. So this way, the optical flow errors does not accumulate, and since the estimation is done independently for each target frame, the direct matching approach can recover after failure. So failure images are just skipped. However, the direct matching is a harder task due to a change of viewpoint, change of illumination, and large motion accumulated over time. So in the video, there is a failure caused by a significant camera rotation and its recovery of matching on later frames. Okay, apart from the simple chain of optical flows and the direct matching, there are other possibilities. For example, the target point could be tracked for 2 frames, and then followed by optical flow from frame 2 to frame 5, followed by another simple chaining. This could be beneficial in some cases. For example, if the frames 3 or 4 are occluded, or there are some other difficulties like motion blur, which caused inaccuracy of optical flow in these frames. But this is not the only option. There are some other options which could be possible too. So number of possible chains grows fast with the increasing number of frames in the video. Note that the best tracking result could be achieved using a different chaining scheme for each query point. So each query point could have a different trajectory or different skipping over some frames. Now I will describe our MFT method, which significantly reduces the number of considered chains and selects the most reliable one. So let's go MFT. Let's go over MFT tracking example frame by frame. So the MFT selects the simple optical flow to the first frame because there are no other candidates. But in a second frame, there are 2 flow chain options. The first one is matching directly from reference frame to current frame. And the second one is computing the optical flow between frame number 1 and frame number 2 and chain it with results from frame number 1. So there are 2 correspondence candidates. And MFT selects the most reliable one according to the strategy I will describe in a moment. Any other candidates are discarded. So in the third frame, MFT considers 3 chaining options. The results on a previous frame chained with the optical flow between frame number 2 and 3. The results on a frame number 1 chained with a direct match from frame number 1 to number 3 or current frame, and direct match. Again, MFT selects the most reliable candidate. This is repeated on each frame of the video. MFT only uses flows spanning 1, 2, 4, and direct match. This selection of logarithmically spaced optical flows or skips significantly reduce the computational cost, making MFT fast. Yeah, and again, the whole procedure is done independently for each tracked points. So each tracked points can have different frames, which is, or the trajectory can have different frames, which is constructed. Okay. So how to select the most reliable candidate? So we estimate uncertainty of each optical flow vector and select the candidate chain with the lowest overall uncertainty. To compute the overall uncertainty, we sum the uncertainties of all flows in the chain. Nowadays, typical optical flow methods extract features from both input images and construct a correlation cost volume, which is then used as the input to the optical flow regression head. In MFT, we extend this with a small uncertainty estimation CNN, which output an uncertainty score for each optical flow vector. We train our uncertainty head on optical flow data sets using a pretty standard uncertainty loss. So during training, the network can balance a high error of the optical flow by predicting a high uncertainty score sigma. However, to avoid zeroing out the loss by predicting high sigma everywhere, the loss contains a regularization term. So standard optical flow methods are trained to work even in occluded regions, which is possible thanks to the surrounding context. In the top right example, the estimated optical flow is correct and has a low estimated uncertainty, but MFT cannot continue tracking this point as it would drift from target point on a head to the occluding bamboo. Because of this, we extend the optical flow network with another small CNN that is trained with binary cross entropy to estimate occlusion maps. In MFT, the candidate flow chains that contained any occluded flow vectors are removed from the candidate pool. Okay, since there is no benchmark for dense tracking, we evaluated MFT on a standard sparse point tracking benchmark called TABWIT. And MFT performed competitively to concurrent work, which are specialized in sparse point tracking while being orders of magnitude faster. The slowest part of the MFT is computation of optical flow. So for application like video editing, the optical flow can be pre-computed, which results in tracking speed of over 100 frames per second and could be used for interactive use. Here's an example of video editing using MFT. So we start with the original video. First, we edit the initial frame of the video, and then we propagate these edits through the entire video using the estimated position outputs from MFT. Here's another example showcasing MFT's capability in handling non-rigid motions. It can handle these motions without restriction on any particular target particle. In this instance, MFT successfully recovers tracking even after a significant occlusion. Okay, I can skip this one. And finally, in our last example, we demonstrate how we change the video style using just a simple example frame of a new style. We then apply this stylized frame throughout the entire video using MFT. So, moreover, MFT can track multiple referenced images. So we are not restricted only for a single reference image. And in this example, the artist created versions of stylized image for 3 referenced images. And we tracked these images throughout the entire video in parallel. We used a normalized uncertainties from individual tracks. The colors correspond to individual tracks. And we use these normalized uncertainties as blending coefficients to create a stylized version of a video. note that this output is not intended to be a final version, but rather input for some in-painting algorithms. So another application of dense point tracking is tracking soft tissues during endoscopic operations or endoscopic surgeries. The images displays MFT tracks on surgical tracking dataset. It is worth noting that MFT has been chosen as official baseline by the authors of the Surgical Tracking Challenge. Surgical Tracking Challenge. And so thank you for attention. All raw results and the code, including a simple demo used to generate these videos, are available online. So thank you for attention.
Thank you very much, Michale, for this very nice presentation. Very nice and very impressive work. So we are moving to the question session. We do have some time for questions. So are there any questions in the audience?
Any questions here?
Thank you very much for your talk. And I have a question about certainty quantification method. Yeah, so my just question, why did you decide to use such type of methods like applying CNN to predict variance? Did you try any other ways to quantify uncertainty?
Actually, yes, but I cannot talk about it because we have just a paper in a review. So yeah, so let's say that the newer version do it differently because variance is not very good for estimation. Actually, it doesn't make sense for high errors in optical flows because let's say if the optical flow error is like 30 pixels or more, the matching network, it cannot have this information, like error above certain levels are impossible to estimate as uncertainties. Like if it's something 2 pixels from the correct position, yeah, we can estimate it pretty much correctly. But if it's 300 or 30 pixels, it makes no sense to differentiate between two. So yeah, we tried a lot of different things, but in that paper is this one. No. Yes.
Okay, thanks.
Thank you. So it's just maybe to follow up on this. So it seems that there is some kind of a robust estimator is needed for the errors. Is that you need to have some dealing with outliers is really what is needed in this case?
Yes, that's true. So yeah, maybe choosing the most reliable optical flow based on this, it's, let's say, working pretty much correctly, but the hardest part is estimation of occlusion, so the outliers which are not visible at all. As you can see, for example, on this particular example, the grass look exactly same, or at least for optical flow algorithms, if it's left or right. So when there are a lot of visible points which are correctly estimated, it's fine. But suddenly when the large portion of the image is occluded, but the newly discovered areas or newly observed areas are very similar to this, the occlusion network has problems to identify if there are outliers in this or not. So
Thank you. Any more questions from the audience?
Yes.
Excellent.
Thank you once again, Michael, for your report. It's very interesting that you managed to track all the points on the image, but can we reduce the computational complexity by using only several specific features instead of all points using this method? Or it's inapplicable?
No, it's not applicable because, okay, the other methods do actually tracking of selected points, like hundreds or maybe even less. However, this particular work is based on optical flow or extending optical flow, and optical flow networks estimates correspondences for all pixels in the images. So we cannot choose just a subset. Yeah, we can specify what are target points, and this provide as output, but it doesn't really matter because the method still has to compute all the points.
Indeed, I understand that it has involved like all the points, but if we need like more interested in one subset of points than the others, for example, to track some specific object, which is hard to distinguish even like for the longer term than long-term tracking.
Yeah, you can combine that with some masking. I don't know, for example, Session-2 has pretty interesting results. So you can mask the object and just track, for example, the lioness on this image, and doesn't focus on the background at all. However, the algorithm which we used for computation of optical flow still will compute all pixels or pixels, all correspondences between images. So yeah, you can specify it for subset, but you cannot, at least with this approach, you cannot save a computational time on this. However, as I showed before, we are still significantly faster, like orders of magnitude than the fastest methods available at the time. So it's, yeah, it really depends on specific use case.
And maybe just to quickly follow up on this. So this number which you quote on the computational results. So this is this 2 FPS. This is including optical flow computation. So this is from scratch if you run it on the video.
Yes,
right?
yes.
And if it's computed, it goes up to 100 FPS. That's what it's saying.
That's true. And new version, maybe it will be next month on archive. It's even faster and more precise. So so this is including optical flow computation.
Okay.
I think that the newer version has better results, and it's like 6 FPS without pre-computation.
Okay, wow. Okay, these are very interesting, very impressive results. So thank you very much, Michale, for joining us today. And thank you.
Thank you for having me. And
thank you very much. I had this very short question, but maybe I'll ask.
Oh, so now, Rostislav, if you have a, please ask. Yeah, I think we can squeeze in one more.
Yeah.
Can you reformulate this approach through considering multimodal particle filters or something like that when you have multiple modes in your probability distributions of possible tracks?
Not sure, but I think we completely skip the probability at all. So now we are not focused on a probability or uncertainties anymore. However, in this approach, yeah, not sure. No.
And maybe
Maybe we can move it to offline discussion, but thank you very much.
Thank
Michal.
you.
Thank you.
Much from the Lviv audience. Thank you very much, Michal.
Thank you. Thank you for all.
So we are moving to the next presentation. We can thank Michal again. We are moving to the next presentation, and the next talk will be given by Denis Herasimuk. I see he's preparing on site. Oh, perfect.
Can you hear me?
Yes. So we can hear you. We don't see the slides yet.
Yeah, I'm sharing 1 second.
Oh, perfect. We see the slides. Excellent. So Denis is a senior data engineer at EPAM Systems at a company, and also a visiting research fellow at the NYU Tandon School of Engineering. And he's going to talk about his work on model auditing and responsible model selection, which, as we all know, it's a very important problem and timely in these days. So Denis, it's great to have you give a talk. The stage is yours. Thank you very much for joining us today.
Okay. Yeah, thanks. So hello, everyone. As I said, my name is Denis, and I'm research fellow at the Center for Responsible AI at NYU, and also a participant at the Responsible AI for Ukraine program. Actually, I also added some slides about this program. You can check out this link to see our website. And today I'm very glad to present our paper accepted to SIGMOD 2024, which is called Responsible Model Selection with Virne View, which is joint work with Fala Rivhan and Julia Stoyanovich from New York University. And let's begin with the motivation of our work. So motivation is straightforward. Accuracy is not the only important dimension of model performance. And the reason behind that is that model predictive error can be decomposed into bias, variance, and noise terms. While accuracy metrics can capture the bias term, they fail to capture variance and noise terms. But variance and noise terms are quite crucial thing because they are related to such dimensions of model performance as stability and uncertainty. So model stability measures whether small perturbations in the input cause proportionally small changes in the output, whereas model uncertainty captures the confidence of a model in its predictions. And model's instability and model uncertainty stems from two main sources. The first one is the inherent randomness or variability in the data. And the second one is a lack of information arising from limitations in understanding the system or process being modeled. To better explain that, I added this visualization. So here you can see in red line real function, and in blue dots we can see our training data. So when we have a small variance of our data and these data points are quite close to our real function, so we have actually low aleatoric uncertainty or data uncertainty. But in areas where we have a big variance of our data points, so we have high aleatoric uncertainty. And in areas where we don't have enough data, we have high epistemic uncertainty or model uncertainty. So also in literature, it was observed that ML models can exhibit disparities across groups in terms of selection rates or error distribution. And these disparities actually can lead to unfairness in real world and even to unlawful discrimination against historically disadvantaged groups. But actually, overall accuracy metrics cannot capture a model's potential discrimination against demographic groups. And therefore, it brings us to the importance of measuring error disparity metrics. So it's our standard accuracy metrics, but measured for specific subgroups. And even popular fairness metrics, which were like provided before in literature don't actually capture whether the model is comparably stable and certain for different demographic groups. And this leads to such important metrics as stability and uncertainty disparity metrics. Based on this motivation, we define the next problem for our work. So since measuring only accuracy is not enough for building responsible ML systems, each ML model has at least 4 dimensions of performance, like accuracy, stability, uncertainty, and fairness. So this brings us to the following challenge. So considering the multitude of model types, performance dimensions, and trade-offs, we face the challenge of responsible model selection. So challenge when we need to select the most appropriate model for the context of use. And actually, these dimensions can have trade-offs with one another. Therefore, we need to understand what among these trade-offs we need to choose for our context of use. And in this work, we present the following contributions. So the first contribution is the following. That's our software framework, which is called VIRNI. It measures all these normally important dimensions of model performance, which is fairness, stability, and uncertainty, both overall and broken down by groups. The second contribution is that VIRNI enables data scientists to analyze performance using multiple sensitive attributes, including non-binary and their intersections. The third one is that VIRNI offers diverse metric computation interfaces for in-depth profiling of model performance. And the last one is that VIRNI implements streamlined flow design tailored for responsible model selection. And next, I will explain 2 of these 4 contributions, which solve the following 2 subproblems. The first one is how to measure these metrics, and the second one is how to streamline responsible model selection. So how to measure these important dimensions of model performance? One of the dimensions which we measure in VIRNI is model fairness. In general, the term group fairness means the absence of any prejudice or favoritism toward a group based on their intrinsic or acquired traits in the context of decision making. And there is such things that there is no one definition of fairness. Multiple definitions of fairness exist and actually are necessary, because what is considered fair depends on the domain. So are you providing a loan, or is it related to university admission? So it's actually quite different domains, and fairness criteria for them can be different. So they are context dependent. So it also depends on the legal and social context. And each fairness criteria is expressed via its own fairness metric. So here, for example, we have 3 example metrics: equalized odds FPR, equalized odds FNR, and disparate impact. And as you can see, in general, it's just differences or ratios of our previously accuracy metrics, but for specific groups. For example, here we compute this metric for disadvantaged group, historically disadvantaged group, which can be like non-white races, and for historically privileged groups, which can be white races, for example. And in addition to fairness, VIRNI is able to measure model arbitrariness. And in our approach, we are using bootstrap methods to quantify model stability and uncertainty, since basically we were working with tabular datasets, and the bootstrap method is one of the most popular ways to measure this in the case of tabular datasets. So how it works, it's quite simple. So we firstly obtain M bootstrap samples by randomly sampling the training set dtrain with replacement. Next, we fit each classifier on the appropriate bootstrap sample and apply M-fitted classifiers on the same feature test set dtest to obtain M prediction sets. And in this way, as previously we had just one prediction for one row, using Bootstrap, we will have M predictions for one row. And in such a way, we can get this variance, predictive variance, which we actually will use to compute various types of metrics. So it's stated that using So in such a way, we obtain the predictive variance, which approximates the model stability and uncertainty of a single model being used to make predictions. And based on this variance, we can compute the following metrics. So the first one is called label stability, which is just the difference of the number of models that classify a sample as class 1 and the number of models that classify a sample as class 0. Additionally, we can compute other uncertainty metrics, which one is epistemic uncertainty or model uncertainty, and this is just variance of our model predictions for a specific sample. And also we can compute aleatoric uncertainty, and in this case, it measures the inherent uncertainty in the data, measuring Shannon's entropy. And we incorporate and build the following software framework to measure all these metrics. So in general, the framework splits the whole pipeline on 3 stages. The first one is subgroup metric computation. The second one is disparity metric composition. And the last one is metric visualization. And the process starts when user defines 3 types of inputs: base flow dataset, models config, and config YAML. And these 3 inputs go to our metric computation interface. There is a rich set of various types of metric computation interfaces depending on the context of use. But for example, here, next we apply subgroup variance analyzer and subgroup error analyzer to compute actually all these overall metrics and metrics for different disadvantaged and privileged groups. And as output, we get our pandas data frame, which goes to metric composer. The idea behind metric composer is that it computes disparity metrics based on previously computed metrics for disadvantaged and privileged groups, because as I previously showed, that it's just differences or ratios of our accuracy metrics, and there is no need to recompute the whole pipeline if you need to get just one new disparity metric. And also in such a way, we can save storage and computation time for that. And next, we pass our 2 pandas data frames to metric visualizers. There are several types of visualizers. There's one static, another interactive visualizer, and we actually use them to build our Virni view tool. And to better show what results can we get using Virni, I added here slightly plots from another paper. So in general, what is shown on this plot is we conducted the following experiment. We took different train set sizes and use the same model and use the same test set. And we wanted to look what will be the trend of label stability when we vary these train set sizes. So, and idea behind this plot is that when you increase train set size, your model stability actually increases. And it's quite a logical observation, which there is like proofs in theory, but we could not find any papers that showcase that in empirical studies. And using Virni, we were able to show that. Also, we can see it for trends for epistemic uncertainty. So when we increase train set sizes, epistemic uncertainty or model uncertainty actually decreases. What about aleatoric uncertainty? I can remind that it's data uncertainty. So as you can see, it increases it improves only slightly. The smaller, the better. And next, after specific threshold, it does not change. And the reason is that aleatoric uncertainty cannot be improved adding more data, which is quite well-known fact, but here it's empirically proved using our framework. So the second problem which we solve in this paper is how to streamline responsible model selection. And I need to highlight here is that Virni was not built to do responsible model selection on behalf of the user. And so in general, it just provides a tool how to speed up this process. And the user, knowing the context of use, can apply this tool to make this process a little bit faster. So for that, we built an interactive tool to streamline responsible model selection. It actually generates nutritional labels for our ML models. And it consists of 6 visual components, each corresponding to a step in the responsible model selection pipeline. So the first step of this process is to analyze demographic composition of the dataset, because it can provide some insights why we see such patterns of model performance. So firstly, we analyze data broken down by different groups. Next, we try to reduce the number of models to compare based on overall and disparity metric constraints. So here a user can define what metrics are the most important for us and what ranges of constraints we want to have. And based on that, we are able to find this reduced number of models that satisfy this constraint. Next, when we reduced it, we can now compare them more deeply using such heat maps, so overall and disparity metric comparison. So here, for example, we can compare overall metrics of this like 4 models, and with colors we show what model is the best one. These colors are adapted to what is better for the metric. If the higher the better, so or if it is higher the better, or it's the lower the better. So these colors can take it into account. And the fifth step is to generate actual nutritional labels for the selected model. So here we select one model and can break down its performance by different dimensions and protected groups. And finally, we generate summarized performance of a selected model across different dimensions and protected groups. We can set specific constraints, and if model cannot be in that range, which is enough for our context of use, it's marked in red. And based on our previous contributions, we see the following venues for future work. So first venue comes from the point that actually this bootstrap method is quite computation heavy process, and therefore we want to speed up measurement of model stability and uncertainty. And we have one ongoing project in this direction, which is called Virni Flow. The second one is that actually in benchmarks which we have in fairness literature, there are no benchmarks that also try to measure not only accuracy and fairness, but also look at stability or model arbitrariness. Therefore, we create our own benchmarks to assess the impact of various ML lifecycle stages on model stability and uncertainty. And also we have one ongoing project in this direction, which is called Shades of Null. And the last one, it's quite tricky problem because we want to integrate multiple dimensions of model performance, like fairness and stability, into hyperparameter optimization step. And it's quite not easy thing if you want to do something smart, but we still think that it's important for responsible model development. And that's quite all. Thank you so much for your attention, and I'm happy to take any questions.
Thank you very much for the very interesting talk. The work is very exciting. I will maybe again start if there are any questions in the audience. I don't quite see the audience, but maybe Rostislav, you can moderate the questions discussion.
Any questions, please?
So my question would be, you used Bootstrap to evaluate the performance of the models. Is there a specific reason to do that? Or, for example, one could also use cross-validation or repeated cross-validation. Would I have a small follow-up question then on that?
Yeah, so idea here is that before applying Virni, you actually why we use Bootstrap approach? Because Bootstrap approach is one of the most used approach to quantify model stability and uncertainty in context of tabular datasets, which are the most popular for fairness literature. And therefore we apply Bootstrap here. And the way how you can work with Virni is that you firstly tune your hyperparameters, for example, for accuracy metric. And after that, I can show on this slide. So it's like trained model with already, we find the best hyperparameters for example, for F1. Next, in Bootstrap method, we already set the same hyperparameters for different estimators in our Bootstrap. And using Bootstrap approach, we actually can compute model stability and uncertainty because it provides us this variance, which we can use to compute such types of metrics which you provide here. So the answer will be that but there are other techniques to compute uncertainty, and the thing is that they are more tied to deep learning. But in fairness literature, we have more tabular data sets. Therefore, we started with the Bootstrap approach, but of course, we want to try other approaches and speed up our Bootstrap approach.
So so you did perform the hyperparameter optimization before the Bootstrap? So,
yeah.
Okay, but isn't it possible that you get a little bit of an over-optimistic performance estimate because when you perform the hyperparameter optimization on the whole data set, then perform the Bootstrap samples and then use the optimized parameters on these Bootstrap samples, it could be that the performance is slightly overestimated. So no.
Yeah, I understand what you're talking, and therefore model arbitrariness should took into account during hyperparameter optimization. Therefore, we define it. It's just interesting future work. But yeah, it's quite correct.
Yeah. So one problem with Bootstrap is also that there are the same observations occur several times in the Bootstrap sample. And so oftentimes one recommends to perform subsampling instead of Bootstrap, choosing Bootstrap samples, because this does not happen then. But yeah.
But we actually doing subsampling here. So actually, this Bootstrap samples took this like 80%. Or each time we take a random 80% from our original trained data set.
Just the last comment, but in Bootstrap, for example, the same observations. So you draw in the observations, and in these Bootstrap samples, the same observation can occur several times. That was I meant.
Yeah. You can actually vary the percentage of how many you want to take into account for each Bootstrap. It's like parameter invariant, but yeah, it's quite cool.
Key things.
Thank you. Any further questions from the audience? I may have a very small question. You started with fairness, and that means that the result of the model should be independent of these sensitive traits. Yes. But on the other hand, you should ensure that your sample is representative. And typically, one just takes marginal distributions or several traits, but that's probably not what one has to look. Did you think, or did you ensure that the joint distribution of these sensitive traits are preserved in?
Yeah, so I understood what you're talking. So in general, Virni is just model profiler. We are not doing any type of optimization or tuning there. And so we just compute metrics based on that model that user provide to us. And as you said, in fairness domain, we cannot use sensitive attributes for training. You try to avoid them. In general, it's like you try to avoid sex and race columns in your dataset during training. But the thing is that it's not always help because we can have some feature covariance between feature correlation between features, and some other features can show that this for example, row is more likely that this row is related to male or female. And the thing is that in fairness, there are three types of fairness enhancing techniques which we can apply. It's pre-processing, in-processing, and post-processing techniques. And pre-processing techniques is when we try to change our data. Specifically, for example, it's like disparate impact remover. When we actually change our input data so that it satisfies a disparate impact metric, and after that we pass this data to our model. Another type of fairness intervention is when we have in-processing intervention. So it's actually when we change our loss function. So that we try to incorporate fairness criteria into our model's loss. And this last one is post-processing. Is when you, for example, your model tries to find like top 10 people for which we want to provide tutoring. And in this case, when model predicts 10 different, makes these 10 different predictions, we apply post-processing intervention, and it tries to so that, for example, in context of fairness for like sex, it tries to make that we try to select 5 males and 5 females. So it actually changes what predictions model did. So it's 3 types of interventions. Which
Thank you.
Thank you very much.
I guess there is no further questions from the audience here. So
I think we are almost done. I'll ask maybe one more. So the software is available? Is it in a web server, or how is it available? Can you say a bit more about this?
Yeah, First of all, it's pip installable. Second, there is documentation. Third, our NYU lab is using this software to compute this metrics because I try to make it very, very flexible. It supports 4 Python versions. It has integrations with the most popular software frameworks in fairness, like IF360. Also, you can profile PyTorch tabular models, which are actually neural networks, which could be applied for tabular data sets. So it's available, pip installable, and yeah, please use it. There are links actually in our paper. So yeah.
And maybe can you say what type of classifiers can you handle, just so that people understand, if it's applicable to their problem?
Yeah, currently we work with only binary classification because it's the most popular in fairness, but we can extend it obviously to multiple class classification. And what types of classifiers? It's scikit-learn models, it's LGBM, it's, as I also said, it could be some models from PyTorch tabular, which is the most popular framework for neural networks on tabular models. So this actually quite good range of models which you can have.
So there is no, actually in the model, there is no assumption on the classifier form itself, right? It can be linear or non-linear, any type, because you just need to be able to train it on different subsets of data.
Yeah. And the idea of ERNI is to be very flexible to compute uncertainty and stability for anything that can be fitted and can return labels and probabilities. I was able to profile fairness in processors when actually loss function was changed. I was able to profile even such joint cleaning and training methods, when during training we also perform cleaning, which are actually quite hard models to profile, but Virni was able to do that. So yeah, we try to be very, very flexible in that sense.
Thank you very much for the very nice presentation. Thank you.
Thank you.
Thank you.
And with that, we are moving to the last talk of this session, which is also very exciting. So we are going to have Dmytro Fishman. Just to say a few words about him, Dmytro is a lecturer in artificial intelligence at the Institute of Computer Science at the University of Tartu in Estonia. He is also the co-founder and chief science officer of Better Machine, a medical imaging startup that is building intelligent tools to enable doctors to diagnose cancer more efficiently. And in his talk, he's going to talk about this interplay of taking state-of-the-art models and putting them into sort of working system, which I think is a very exciting topic, and I'm really excited to hear what Dmytro is going to say. Dmytro, we are very pleased to have you here today. This stage is yours. Thank you very much for joining.
Thank you, Joseph. I think I would need your permission, or somebody who is the admin of this call, to share the slides. Otherwise, I can't share the slides. Oh, now I can. So thanks. I'll do that just in a second. I hope you can tell me if you see my slides. From the Lviv people, could you?
Yes, everything
right. Amazing. Thank you very much, Rostislav. Right. So first things first, I really enjoyed the previous talk. I'm happy that I was able to join a little earlier. So I was really fascinated to listen about hyperparameter optimization, especially when it comes to such difficult things such as fairness, which is super hard to define, right? So thanks, Denise, for this. And also, I'm very grateful to the organizers for inviting me. Yes, indeed, I am a lecturer at the Institute of Computer Science. From the next week, I'm going to be associate professor of the artificial intelligence at the Institute of Computer Science. I'm also invited lecturer at the Ukrainian Catholic University, and also co-founder and chief science officer at Better Medicine. So we are basically building AI models for doctors. That's short as it. But I think the most important affiliation that is relevant for this talk is that I'm head of the Biomedical Computer Vision Lab. So this is the talk is going to be about projects that Biomedical Computer Vision Lab was having over the years that is centered around biomedicine, and the papers that we have been basically research that we were having over the years. So I'll give you a set of selected research projects that me and my lab we were pursuing over the years, and I would be absolutely delighted to share as many details as I can about these projects. Before we delve into details of these research projects, I usually give a short introduction of what is this field about. And when I tell about biomedical field and research on cells and cancers, it's good to put people into context of what scale of a problem we are talking about here. And when I'm usually in the audience, and I'm very let's say I'm very sad that I can't be in the audience. I would love to be in the audience there in Lviv. I missed you guys so much. I usually ask people a question such as, how many stars do you think there are in the galaxy? And usually people don't know the answer, and I'm really glad, because that would mean something is not right. And I usually tell them it's about 200 billion stars in our observable galaxy. And then I ask them another question, which is, how many do you think we have cells in average grown human body? And that also takes quite a bit of time for people to figure out. And unless they're Googling, they don't know. And the answer is about 37 trillion cells. So it's actually trillion cells comparing to 200 billion stars. So the scale of the problem, when you think about studying cells in a human body, it's a lot. It's much bigger number when it comes to if you think about galaxies and stars. Also, comparing to stars, our bodies are a lot more intricate. They're parts, cells that communicate in so many interesting and non-trivial ways. So there are a lot of things that is happening in our bodies. So it's basically these trillions of cells that make up tissues. There are about a dozen of different tissues. And these tissues make up organs, and all that machinery is there to help us live happy and healthy lives. And as you might have followed my talk so far, this system is very complicated. It's very complicated. It's very subtle. It's very fragile as well. So there are a lot of things that can go off, that can go wrong in this system. And one of these ways how things can go wrong is that, we usually throughout the lifespan of a human, we tend to acquire mutations in our DNA. And some of these mutations, they cause the cells to divide rapidly and uncontrollably, causing this division to invade the tissue where the cell was initially residing, and then forming this kind of lumps in the organ. These lumps tend to grow and send their messengers to other organs, practically invading other organs, the process which is called metastatic spread. And this metastatic spread essentially leads to complete demise of a person, complete seizing of life, basically killing the patient. And this is a very complicated problem that I'm illustrating. So this is a story of cancer. If some of you might know, some of you might recognize, this is how cancer is developed, and this is what happens when the patient is sick with cancer. And now I'm coming back to the computer science part of my title of my talk. So my title of my talk is about computer scientists making progress or making difference in medicine. And us as a computer scientist, we have been not really solving that sort of problems, as you can see on the right side of the slide, but we have been more thinking about other things. So for example, what Denise was just explaining, for example, we were rather successful building great classifiers for, let's say, images. We have been rather successful taking most of the control of the drivers in the cars. We have been rather successful building models that can draw or can pretend to make art. So we were pretty good as computer scientists. We were pretty good at these sort of things that you can see on the left. So as it turned out, and this is what I have figured out throughout my career, is that there is a link between those things that you can see on the left and those things that you can see on the right side. So it turns out that some of these problems that we as a computer scientists were able to solve using computer science approaches, and most likely that I'm talking about AI right now, or deep learning for that matter, we were able to solve some of these problems on the right side of the slide as well with the same type of technology. And I'll give you a couple of examples on a higher level, and then I'll go into more specific projects as we go. So what we can start with is cellular level. So I just explained to you how cancer goes from cellular level to tissue level, and then it starts to invade the organ level. So it becomes visible at the organ level. But first thing, what happens is the cellular level. So this image that you can see on the slide is actually an image of a cancer cell line. So this specific image, these specific cells, they are cells, by the way, they are cells, and they are cells of cancer. I do not necessarily right now can tell you exactly which type of cancer it is, but this is how cancer cells look like. And what usually happens when somebody is developing a drug, let's say, that's supposed to fight cancer, is that they don't right away recruit volunteers, human volunteers, or even animal volunteers. They don't recruit anyone. In fact, what they do is that they study the way how their drug is performing on cell lines. So only on this kind of artificial, in a very artificial environment. So they add this drug to the cell line, and they see how the cell line reacts to this drug. So what they want obviously to see is that if it's a drug that's supposed to kill the cell line, cancer cells, then that the cells would start dying, right? And one obvious problem with this is that cells are very, very tiny, which means that counting and looking at the cell lines by eye is very, very difficult. So you can't really count all the cells. So instead, what we were doing in our lab, in collaboration with a company called Revity that produces microscopes for laboratories that study cancer cells, is that we were training very simple convolutional models to segment and detect these cells, and that would help biologists and lab practitioners to quantify and gain evidence for or against certain drugs that they experiment with. So that is that was the thing which we started with in this field. And then there are very different ways how you can study cells. And the previous picture I showed you fluorescent images, which is just a way to illustrate, to visualize cells. And this image here is much hotter. It's called bright field images. And in bright field images, it's much more complicated to see cells. And we were able to adapt existing approaches for cell segmentation and detection. And we were still able to quantify and detect cells from these more complicated images, which were before that considered to be a junk material, was not useful at all. Now, when we move on a little bit up the ladder of the cancer progression, then we come to the pathology or tissue level. And what is happening at this level is that when people suspect having a cancer, they usually take a biopsy. Biopsy is a surgery type of procedure where they put a needle in a human and they retract a small piece of the cancer, basically of a tumor. So they take a part of the tumor, they slice it up, and they put it under the microscope, and then they study this images. So this is the image that you can see on the left side of the slide. This is how the sort of tissue looks like under the microscope. So it's a little bit higher scale image than what you just seen in the previous slide. In here, we collaborate with local hospitals and local pathology labs that usually do all these operations manually by just looking at the microscope. We help them to detect different structures onto these pathology images, and we help them to make the diagnosis. So we help them to automate the diagnosis performance. For example, with this specific image, we help to detect the structures called tubules and also count cells within the tubules, because that was a very important marker for the disease in that very specific case. By the way, this specific case is not really a cancer case. This is actually a male infertility disease, and those purple things are called tubules, and the cells inside, they indicate whether a human, a male person is there infertile or not. And then we finally come to the organ level research that we do. And this organ level research we do with a company called Better Medicine. As I said, I'm one of the co-founders of this company, and currently at present, I'm acting chief science officer of this company. And what we do with Better Medicine on the CT images, so these are called CT images, we help to build AI models. In our case, we built CNNs and some transformer models to detect and measure various organs as well as pathologies that are present on these images. So, for example, here there is a big pancreatic tumor. So we have liver, we have kidneys, and we have a pancreas. And in pancreas, we have a huge pancreatic tumor that can be detected and measured. And this is what we help doctors to do. We basically do this work for them and give them the final results for validation, for verification. That is a short summary of what I have said so far. Hopefully you were able to follow. So on a cellular level, we are helping to detect and quantify cells from microscopy images of various kinds. On the tissue level, we help pathology labs, and we work with local Estonian hospitals to help to automate this work of a pathologist, somebody who has to look through hundreds of these huge images and understand all the structures and find all the structures. So some part of their work can be automated and can be made more efficient. And finally, on the organ level, we are building deep learning models. We're training deep learning models to detect and measure and quantify tumors. That's what we do. And there are selection of several projects that I wanted to highlight, so that either have resulted in publications or are very close for publications, and that I think are most interesting. So the very first project, and just a reminder that we were using this kind of bright field images, and we were detecting nuclei from the bright field images. And as I probably also mentioned, is that the bright field images, so this is a bright field image, it's very hard. If you look carefully, you look long enough at this image, you might understand why is it hard. Is that there is a very low contrast, there is very little signal present, and signal noise ratio is very skewed. And although there is an advantage to these images, and the advantage is that it's very easy to acquire. It's very easy to get these images. Before the deep learning revolution, these images were considered to be junk. They were considered to be non-useful, because humans could not work with these images, neither could classical algorithms, classical computer vision algorithms. And partially you can understand the complexity if you look at the overlay of the fluorescent images. So this is an overlay of the fluorescent channel with the bright field channel. So you can see the centers or nuclei of the cells, which you could not probably see on the bright field itself. But that is something that biologists want to see in their experiments. And using fluorescence is good for detecting cells, but the problem is that obviously fluorescence comes with a price. You have to pay money, you have to spend time to prepare the experiment, and the third side effect is that fluorescence kills the cells. So if you would like to study cells for long enough, then you won't be able to do that with fluorescence. So this is something you can do with bright field, but it's not something you can do with fluorescent images. Finally, so we did make it happen. We did train a model that was able to find nuclei from bright field images. something that humans could not do, but thanks to the interplay fluorescent and bright field images, we were able to train them. And now it's been deployed in production in a big company, and they offer it as a standard offering to their clients around the world. But we didn't stop there. So this was just a part of the plan. Another part of the plan was to detect the whole cell, so the full area, which is covering the cell. And that is a lot more complicated problem. So we were after something called whole cell instance segmentation, which is like a step up in a complexity ladder when it comes to detecting cells. And what we have done here was that, oh yeah, there is one part more, there is additional part in the complexity of this problem is that cell bodies, or we call them cytoplasm, they like to overlap. They are not necessarily completely isolated from other cells, so they do intersect, as you can see here on the slide, which does not necessarily happen very frequently in the natural objects world. So all these big data sets that exist for images of the natural world, you don't normally see a bicycle behind a track, while you can see one cell behind another cell. So this is where the complexity of the biomedical field comes in. So in order to accomplish this task, we actually sat down and our students, they have labeled a massive amount of this kind of cells. So they actually by hand drew the cells to collect this unprecedentedly large data set that includes the cell bodies, cell cytoplasms, which are overlapping. And then at first we trained a very classical UNet model that also worked in a standard bright field nuclei case. But it turned out that it doesn't work. And there are several obstacles or several problems with applying UNet model on these sort of images. And one of the problems is that it's not meant, the UNet, the classical UNet model, which I hope that some of you might have familiar with, is the model that is used for a lot of biomedical segmentation tasks. It's not meant for instance segmentation. At least inherently, it's not capable of doing instance segmentation. So what we have done is we have adopted the architecture. We changed the architecture in a multiple ways, but one of the most important ways is that we added 2 new branches into the model that are actually intertwined. They're not exactly isolated. They're actually interconnected. And one of these branches we call instance branch, and the other one is called mask branch. So the instance branch is what you can see on the top, is the image which is concerning itself only with the particular seeds of every cell. So it's only interested in detecting the, let's say, the cluster centroids of the cells. While the mask branch is something you can see on the bottom of the slide, is basically tasks with the segmenting the borders of every cell, but it does not care about the in particular, it is not distinguished between individual cells, but it just finds the borders of cells. Now, when we combine the two and we add some nonlinearity layers ahead of it, we actually got a very reliable and very accurate cell border prediction from the bright field images. We have developed so far, we have collected our own data set. This is the one that you can see in the middle. We have also synthesized some data set that you can see at the top row, and we have run this model on actually on a number of public data sets. And what we see across the board is that our model seems to be state-of-the-art or very close to state-of-the-art with all the other approaches that we have tested. And it's very, very promising. And this is going to be a paper. So this is called Instance-Aware UNet. And we are finalizing a paper and submitting it to ICTERI 2025. And one of the probably driving people who are driving this research is Jaroslav Pretula. He's in the past, he's a student of Ukrainian Catholic University, but hopefully also in the future. So he's a local boy. It's been a great honor working together with him. And another project related to cells is something that we often do discard, or we often ignore as scientists who work with images, is abundant meta information available about images. So in the world of microscopy images, we always know information about the type of the cells we are working with. We know, for example, how close we are looking at the cells. This is called magnification factor. We know the microscopy type we're working with, and all these extra things that are usually discarded. They're usually ignored, and they're not really paying attention to this. So we set out to investigate whether this can be changed, whether this extra information can influence the performance of the segmentation models. And it turned out to be true. It turned out to work. And we incorporated this information through a mechanism called squeeze and excitation. This is something that is rather popular. This is a very popular way to introduce something called attention, something that has gained a lot of popularity recently because of the transformer models. But it turned out that you can do something very similar in the convolutional models as well. I'll not go into too many details because I don't have that much time to explain the details of this project. But basically, the results look very promising. You can see if you look at the top part of the slide, then I have 3 pictures there, and the one on the left is the bright field image with the ground truth annotations overlaid with this. On the middle, you can see the baseline predictions, so the predictions of the model that did not have access to the meta information. And on the right, you can see the model that had access to the meta information. And you can see that in general, it looks more reliable. So the colors are blue is the false negative, and red is the false positive. And what we can say in this is, of course, cherry-picked example. From the quantitative side, it also performing slightly better. What we also saw, if you can see on the bottom part of the slide, is that meta information can impact the way how the model actually performs, meaning that it can actually tell the model to change its behavior. And you can see that on the, again, on the bottom of the slide from the very left, you can see that this is the huge kidney tumor, and its dashed line represents the borders of this kidney tumor. On the next image to the right, you can see in green, this is what the model produces for the mask of the kidney tumor. If you say, model, that the tumor is large, then the next image shows the output of the model if the meta information indicates that the tumor is medium size. And finally, the last rightmost picture shows the prediction of the model if meta information indicates small tumor. So this research has been published at the workshop at MICAI. It's one of the top medical imaging conferences. And actually, another maybe a small compliment to Ukrainian Catholic University is that one of the main authors was Yaroslav Plutenko, who was my student at Ukrainian Catholic University. That was the cell level. We have a lot more projects related to cells, but because I want to show you all the things we have done, we are moving on to the tissue level. And in the tissue level, one of the most complicated problems is the sparsity of the annotation. So because the tissue slides, if you have ever seen pathology images, they are huge. There are thousands, or let's say hundreds, or even dozens of hundreds of pixels in diameter and size. They're very big, and they are very dense in terms of information. So it takes a lot of time to densely annotate those images. And what usually happens is that pathologists they skip some cells when they annotate those images. So for example, you can see that some boxes are there around some cells, but there are many boxes that are not been given by the pathologist. And this is something that usually confuses the model a lot. And the last image here is the annotation that we would love to expect. This is something that we would want to have. In reality, the middle picture is what we usually have in the pathology world. Now, what we have done to remedy this problem is something called generalized background recalibration loss. So in a nutshell, we are telling the model that when it produces false positives, so it's not, let's say, when it thinks that, or let's say when it predicts a cell, but there is no cell in the ground truth, this is usually referred to as false positive. And it's a penalty that that the model takes in as a feedback, as a gradient descent, in terms of gradient descent. But the generalized background recalibration loss, it's actually discounting this false positive error. It says that if the model is predicting very confidently false positive, then I take it as a real signal, because I know that there are a lot of cells that do not have annotations. So if the model is very confident that there is a cell, it must mean that indeed the cell is there. And it turned out to work, as you can imagine. So the left side of the slide is the ground truth. So all the boxes are the boxes from the pathologists, and we asked them to be very, very precise and to annotate as dense as they can. So they did what they could. And in the middle, you can see the work of the baseline model. So the model that was not trained with recalibration loss that we have proposed. And the rightmost column is the column where you can see the results of the model that had this loss in place. And you can see that the model actually performs much better. It generates a lot more boxes. But of course, this is again the visual example. We have, you can check out the paper. This was published as a tiny contribution into ICLEAR 2023. You can find the paper and check out the results in more detail. Finally, we have moved on to better medicine. So this is where we perform organ level research. At Better Medicine, they're taking this kind of full body approach, meaning that they want to build deep learning models for segmenting tumors in all the organs inside the abdominal cavity. This is what you can see is illustrated on the slide. And at the moment, what happens is that Better Medicine has lungs and kidney models in a clinical validation and certification, and liver and pancreas models as a proof of concept models. These are very early results for these two models. Again, as I said, for the kidney model has been rather successfully validated. And the lung model, so the model that is detecting tumors in the lungs, it's been going through the validation process. Now, a little bit more about the kidney model. We call it renal vision. And what it does, it essentially performs semantic segmentation into three classes. And these are kidney tissue, a tumor, or a malignant finding, and the benign finding. So these are the three classes which we are detecting. And once this has been done, once the segmentation has been performed, the next step is to measure and report these findings to the radiologist. So we tell the radiologist, hey, we found this many malignant lesions, this many benign lesions, and this is their sizes. Because this information is very important for the clinical decision-making. As you can imagine, medical community will not accept anything that is not proven to work reliably, and therefore we have run and executed a special clinical investigation in which we have validated this model. In this investigation, we employed 6 local radiologists who have examined 200 CT scans, both with the help of our model as well as without our model. There was a washout period involved so that we can actually have more data for the study. And we have randomized the study. So basically radiologists didn't have the same order of the study, so they couldn't really communicate with each other. So all sorts of biases have been taken into account. And what the study has shown is that we increase the productivity of the radiologist by, on average, 33% and up to 52%. So this is the speed up of the radiology workflow. Second thing, what we have shown is that we slightly increase the patient level sensitivity, which means that they are capable of finding people with kidney cancer and more accurately. And finally, we have increased the interradiologist agreement rates, which means that they would agree more frequently with each other about the correct diagnosis, about whether a kidney lesion is malignant or benign. So this study is being finalized and will be soon submitted to the journal, aiming at Nature Oncology. But of course, the editors will always have their say, and of course there might be changes in that plan. Why we do measuring lesions is because we would like to figure out the dynamics of the cancer. And what happens when people get diagnosed with cancer is that they are usually invited for a follow-up scan. They come for the follow-up scan, and then the radiologist has to examine both the original scan and the follow-up scan. And this is where our kidney model would come in. It would segment both scans. It would identify all the lesions in those scans. And then what we do is we do something called alignment. So we register the two scans so that we can understand which scans come from how to track the lesions. And once we align them, once we register the scan, we can quantify those lesions, we understand their volume, area, and diameter, and then we produce a comprehensive report for the radiologists for their decision-making, so that they can use it for the decision-making. The problem is that it's much harder to do than it sounds. So this registration is very complicated because people are changing over time. They change their posture, they gain weight, they lose weight, and therefore registration is very complicated. So recently we have applied and received funding for building an AI-based approach for estimating something called a deformation matrix, where we built basically a tensor that helps us to understand which voxels from first scan correspond to voxels in the second scan. By the way, I wanted to ask if I have a few more minutes, or is it that I have to cut my presentation short? I have like a few slides left.
Yeah, please continue. Please continue. We started a bit later. So.
Yeah. I just wanted to make sure that that's all fine. Right. So that is the story about the project related to dynamics assessment. So with this, what we do is we track lesions over time. And with this deformation matrix, we understand how lesion has changed in the past. How has it been in the past, and what is the current state of the lesion? Finally, two last projects, which I think are very, very interesting and very important. And one project is related to something called counterfactual in painting. So I'll try to explain. Here, what you can see on the left side of the slide is the CT scan with the kidney cancer. So there is a kidney cancer in the circle, in the red circle. Now, just a bit of a context here, that one of the most expensive and one of the most complicated parts of being in biomedical research is data annotation and data in general. There is a lack of data, and there is a even harsher lack of annotated data. And data annotation in this field is very problematic because it involves hiring very highly skilled professionals who earn a lot of money. So you have to pay them a lot of money, and it also takes a lot of time. So instead of, so basically training full-fledged deep learning models that require pixel level or voxel level annotations is very complicated. So instead, what we're trying to do, weakly supervised approaches. So this is an example of the weakly supervised approach, where we say, okay, what if we just say that the scan contains kidney cancer? Can we make use of that information? Because this information is relatively easy to come by. So what we do here is we train a classifier that can tell whether whether the scan contains kidney cancer or it contains a healthy individual. So you train that kind of classifier. Now you feed the prediction of this classifier into another model, which is called Generative Adversarial Network, GAN model. And the purpose of this model is to generate an alternative version of the scan such that the classifier will be tricked. It will not be able to predict the correct class from this alternative version of the scan. And it turned out that the only way this can happen is to GAN to generate an image without kidney cancer. So it generates, as you can see here, this is an image with a second image with a circle. It generates a scan where there is basically a hole instead of kidney cancer, right? So when you subtract the original image from this generated image, you get the mask of the tumor. This is where the tumor is located on the scan. And this is fascinating because if you think about it, we just started with an idea that I'll just tell you that there is somewhere a kidney cancer on the scan, but I'm not telling you where. And then the whole approach learns to find where on the image is the kidney cancer. And I warn you that the kidney cancer is a very complicated thing. It can take any shape or it can take any morphology. So it's very complicated. There are a couple of examples how this approach works. In the red contour on the left is the input image, the middle image is the counterfactual, and the difference map is what we basically refer to as the location information, is what you can see on the right. And there is another example of the same sort. And the final project that I wanted to discuss, and very quickly though, is using latent diffusion to generate more conditionally labeled data. So again, the same problem which we are facing is the lack of annotated data in the field. And one way to get more annotated data is generate it. And the way how we generate this annotated data is through conditional generation. So we provide a mask of a tumor that we sample from the public available data sets. We add it to the scan of the healthy person because most of the people that come and get their CT scans done in the hospital do not have anything extraordinary on the scan. We put it on the scan, and then we add it as an input to the latent diffusion model that is producing a scan, which now in this place where there was a mask, it produces a kidney cancer. And now what we have is we have a scan from a hospital with a person that has kidney cancer in a place where we wanted it to be. And this is obviously without any people being harmed in the process, right? Nobody got their kidney cancer, which is great. But instead we have a scan where we have a mask and we have a corresponding image. And we can now train our AI models. We can train our UNet models to detect these. And these are just a couple of examples here. So again, on the left, you can see the input image with the white mask that is just being placed there as a placeholder for a model to fill it in. The middle column shows the kidney cancer being generated by the model, and the right image shows you the actual scan, so you can see what was there underneath the mask. So there was no kidney cancer, as you can see. The same is for the other example. This one is a bit more pronounced, and you can see that the model was able to generate kidney cancer there. We could do a very similar thing with the basically generating healthy examples instead of, so if you have a person with the kidney cancer, you can actually generate healthy example by just occluding kidneys. This was a very interesting finding of that project as well. And finally, because I don't want to test the limits of my time availability, I'm wrapping it up, just saying that what we have talked so far is that on my lab, we are doing research in biomedical imaging field. We work with images of cells where we detect cells, we quantify cells. We work with tissue images where we help pathologists to automate parts of their workflow. And finally, we build deep learning models to detect and measure and monitor tumors over time in CTs. So that's all from my side, and this is my lab. The numbers in the bottom are already outdated, and this is the company. So if you wanted to see, these are the real people behind the slides. Thank you.
Thank you very much, Dmitro. It's a very impressive work.
Thank you.
Very nice and very nice presentation too. Thank you. So we are a bit over time, but let's still, have a few questions. But I will pass to Rostislav if there are any questions from the room.
Yes, thank you, Joseph.
Yeah, so thank you for your talk. And I have a question about, you showed models that you built and you tested them. It seems they quite well. But for me, it's interesting, what types of tests should you pass to push your model to production, especially in such field as like healthcare? I know it's quite hard, it's quite long to explain, but maybe you can provide some grasp what types of requirements they put to you to push your model to production.
Thank you. I think it was Denise. No, if I'm mistaken, who gave a previous talk, but maybe I'm wrong. Thank you for a question, by the way, because I can't see you that well. I'm only seeing like a bigger picture. I don't see the details, but indeed, the path is very long and full of horrors. I can tell you that. So one of the probably most important requirements is that company that wants to deploy these models in real hospitals and actually treat, or let's say in our case, diagnose real patients, they have to make a clinical investigation where they have to show with real data, with the hard data, that indeed their whatever system that they are trying to implement works. So because we are not a class 3 device, so we are not interfering with human liquids or tissues, then we didn't have to do it in multiple trials. We did one study with a real radiologist in a real clinic. So this is what I'm showing here. This was done in real clinic. It was not in a way fake or made up. So this was in a way real people who work in a real hospital. And the data that you collect, it has to be convincing enough. So it has to be significant, obviously, with all the tests being significant. You have to show that you have avoided all the major biases, such as what about your gender distribution? What about your so all the fairness questions that if I understand correctly, who's asking a question, all the fairness questions are very, very relevant here. So for example, we have had half of our population of the scans coming from females, half of them coming from males. There was an age match. All these things have to be taken into account in order to convince multiple reviewers that indeed you have done everything correctly. Before that, you have to go through something called a QMS. This is a quality management system where you show that whatever you do in the company is of a good quality. Then there are ISO standards, which is international standards of software development, AI development, all these things. These are in parallel to the clinical investigation. So clinical investigation is one hand, and there are all this ISO, QMS. Then there is a final audit of the committee, which looks at all the data that you have provided, and they give you lots of questions. Then I think we have spent like half a year, I think, only answering questions. And finally, we are right now at somewhere close to getting the CE mark.
Thank you.
Thank you. Any more questions? Rostislav, please.
Thank you. So I have a question related to if you follow up performance or progress, how your technical approaches influence doctors, because, for example, in my understanding, it can raise like technical bias. If I'm a doctor and I have help from a technology, it may later influence my decisions. Do you follow such metrics or something like this?
Yeah. So when when you convince reviewers that indeed it's going to be fine, trust me, it's going to work, then they always say, no, we don't trust you. It's not going to work. They ask you all these questions. So they ask you, what about, biasing radiologists and that and this. So indeed, we have seen it. Even in the study, we have seen that some radiologists trust the model. That's it. And actually, it's a little bit something that shows through this graph. It's a rate of a speed up that basically from the start of the study all the way until the end of the study. And you can see that the rate of the speed up is increasing. And one reason why it is increasing is that more and more radiologists started to trust the model because they saw that the model is making sense. And one way to mitigate the this overtrust or the technical bias, as you called it, is to inform. Is what we have in our intended use in all the documentation is to say that you are the final responsible. You are the person to be responsible if something happens. Then the other thing which we do is we do put all these warnings that, are you sure? Have you looked? Have you checked? Is it all right? Is it okay? And the final thing is that, of course, we are relying on radiologists to catch up, to prevent bad things from happening. But what we have to do ourselves is to ensure that the model is performing the best possible way. And even if the pilot is drunk, the plane should land safely.
Thank you. Any other questions here? I have a small question. You showed 2D segmentation of malignant tumor, but isn't also interesting to build a 3D segmentation? It may give more information for doctors about the size and location.
You're absolutely up to the point. And actually, all the segmentations I showed are 3D, all of them. And the reason is that the problem is that slices are very similar. Nearby slices are very similar. They're almost 99% similar sometimes, depending how thick the slices. And if you train a 2D model, then for each slice for a 2D model is like a new image. And this is obviously not the case. It has to take into account all the surrounding. And that's what 3D models are meant for. And this is another problem because 3D models are harder to train. And but that's what we do. We do 3D.
Yeah, thank you. Yes, we have another question.
Thank you for the talk, for the great talk. One small question. So in one instance, you generated images using a deep learning model, I think, and then so images which do so images where it is simulated basically that the cancer is there, and then you trained another model to distinguish between the between images where the cancer is there and where it is not there. Did you maybe check whether this behaves in the same, so whether the generated images are realistic enough so that the model trained on this artificial data will be reliable then?
Right. So I expected this question. Thank you. Every time I speak about these models, I get this or different versions of this question. Basically, the question is, are you sure? The answer is no, we are not sure. So far, so we are still in a progress with this research, so we are not sure. We haven't implemented anything that would affect patients, and in this case, it's a pure research yet. And so far, I was able to trick radiologists into believing that artificial stuff is actually real. So I take that as one hint that indeed the model has generated something realistic. But what you're suggesting is completely right. And what we are doing right now is actually testing what the model's actual behavior is going to be like. Would it help? Would it introduce more problems? You're absolutely right. There are a lot of questions that needs to be answered, and this is research in progress.
Thank you.
I have like another question. Have you tried any uncertainty quantification methods for your models?
We have not, and I regret this deeply because we should and we will. Well, as long as I am a CSO there, we will for sure. And the reason is very clear. We need to give the estimate. We need to give some kind of basic uncertainty estimate. At the moment, we don't, and that's a big issue.
Yeah, models sometimes can show when they don't know what it's important, seems.
Yes, I cannot tell you how much I think this will improve the user interaction. Absolutely.
Yeah, thank you. There seem to be no further questions. Joseph, do you have any?
Yeah, or maybe just a quick comment on this in the image which you show. I think the good news is that you can actually quantify, what really matters in the performance on kind of held out as data. So I think if you can show that the generated images help that in a kind of a, unbiased and also kind of non-leaked way, I think, that was going to be the real proof. And the synthetic data, it's really storming different fields as well. It's being used in autonomous driving, which is also safety critical domain. There, of course, there is much more training data available in general because, it's, I guess, easier to capture. But I think there is a, in the end, like the real proof is the real test results which you can get. So I think.
Exactly. That's what I keep telling to all the people that you have no idea how many people ask me questions about the synthetic data. First of all, it's very trending, and therefore they're like, they know about it. And yes, absolutely. I think for me, the final indicator is, does the model work? Does it work on all the scans equally good? Yes. And then I'll start looking into figuring out what exactly is wrong and what are the typical problems that it's having. For me, the realism of the generated tumors is a secondary thing, because if at the end of the day, if the model is doing things on the real data or the real patients, then that's the most important indicator. But of course, we are using public data a lot for these sort of things as well.
And it's true that in other domains like robotics, even non-realistic data help the classification, like the output performance, like this domain randomization work with arbitrary textures. I think so. There is as long as the real data is within the kind of, the manifold of the generated, it's okay. So I will sort of not delay this further. Thank you very much for this wonderful presentation and very interesting discussion. So thank you again. Thank you.
Thank you very much. Thank you, audience, as well. And, Slava Ukraini.
Thank you, Dmitro. And with this, let's close this exciting session. I will thank all the speakers again for very nice presentations. Rostislav, do you want to have any final words?
Yeah, I would like also to thank all the speakers of this afternoon or second session in the afternoon, to Michal, Dennis, and Dmitro. And also I would like to thank the main driving force behind this event. Thank you, Vadim, for suggesting these form of participation, and thank you, Joseph, for actually making it done.
I just have to say a few words for the closure of this company. Thank you, Joseph. Thank you, Rostislav, just for making these wonderful event, sub-event at ICTERI true and very useful to the public. Enough audience here, everybody's happy. So thank you very much. I do think that lightning talks at ICTERI have a real future. Very good work.
Thank you. Thank you, Vadim.
So bye-bye and see you next time.
Thank you very much. See you next time. Hopefully in person.
In person in Rio.
Yes.